So today we shall discuss about the review of multisubstrate kinetics. It is opposite or not opposite but a type of substrate kinetics which you have seen previously. If you remember, when we began with the unit number 2, we began with kinetics and we began with a single substrate kinetics. Through using the single substrate kinetics, we have derived what is known as Michaelis-Menten equation, equation. Through equation, we have derived a number of other plots. One of the prominent plots that we know is the Lineweaver plots or the double reciprocal plots. So in any of these plots that we have discussed, we know that what is eventually the Vmax, what is Km, what is slope, and what is intercept. All these concepts by now you should be familiar. When we talked about the uni-substrate, what does it mean? Uni refers to single substrate. Uni refers to single substrate. So to refresh your memory, if you remember when we derived the equation, we began with an understanding of how invertase works upon sucrose, right? So when invertase works upon sucrose, here there is one single substrate is involved. Now people are going on coming and is going to disturb me. That's why I said come in time. Now after 15 minutes people are joining. Okay, so on a single substrate, enzyme called invertase works upon. And if you remember, in the uni-substrate, we have discussed about the three different orders of reaction. The first order, the second order, the zero order, and the pseudo order, etc., and the third order reactions. So in the uni-substrate, it is only one single substrate which is involved. And to this one substrate, one enzyme comes and acts upon. In other words, this substrate will go and bind to the enzyme, and that is called uni-substrate. Now, in the biological system, more than 60% of the known biological reactions, there are more than two products involved. Or in other words, there are more than two substrates involved. So to an enzyme, not one substrate, but two substrates are going to come and bind, eventually getting converted into two products. So that is what's seen here. So there are two products here, two substrates here, A and B, binding to the enzyme E and resulting in the formation of the two products, that is P plus Q. In this bi-substrate, that is two substrates, as we talked about, it is also known as the multi-substrate. Okay, so it is either two or more than two. So that is called multi-substrate. So the kinetics that we'll be studying about in this perspective, that will be known as multi-substrate kinetics. Here, the functional group called X is transferred from one substrate to another substrate and eventually leading to the product formation. This is what is happening. So what is happening in these bi-substrate reactions? There is a transfer of functional group. So if you remember, the functional group here is X. This X, with the catalytic action of enzyme, is transferred to another substrate called B. So this is one kind of an action reaction that happens, mechanism that happens in the bi-substrate reaction. On the other hand, you can also have oxidation reduction reaction. So this also can be possible, oxidation reduction reaction. An example for this would be hydrolysis of peptide bond by trypsin is the example. Hydrolysis of peptide bond by trypsin. In this, the transfer of peptide carbonyl group from nitrogen atom to water occurs. So we shall see this here. And the example is given here. You have the peptide here, hydrolysis of peptide, where this carbonyl group is transferred to the water molecule. So this is one kind of a transfer that happens when the two product or two substrate involved in a reaction. Another example for this would be is the alcohol dehydrogenase, where the hydride ions are transferred to the redox potential like NAD+. So these are the two examples given how there are two substrates involved and that results in the two formation of two products. So although such bi-substrate reactions could in principle occur through a vast variety of mechanisms, only a few types are commonly observed. So in biological system, as I said, there are mostly 60% of the known biological reactions belong to that of the bi-substrate kinetics. But only few commonly occurring is observed. So in this session, we now decide, like what we have done for the unisubstrate, what could be a plot can be applied for the two substrate kinetics, and what could be the slope, what could be the Vmax, what could be the intercept is what we are going to see in this session. Any clarification?
No, sir.
All right, thank you. So we move to the types of bi-bi reaction. Now we are introducing a term called bi-bi reaction. So far I've used the term two substrate kinetics, correct? And now we are using bi-bi. What do you mean by bi-bi? Any reaction which involves the two substrate resulting into two product, they are known as bi-bi reaction. So and that is what exactly is meant here, right? So there are two substrates leading to the formation of two products. So this is called a bi-bi reaction. So when you talk about the bi-bi reaction, it has got two types. What are the two types? The first one is the sequential reaction. Second one is the ping pong reaction or the double displacement mechanism. When we say sequential reaction, it is a single displacement. We shall talk about displacement little later on. What do we mean by single displacement? What do we mean by double displacement? In terms of a sequential reaction, there is single displacement. Okay, there is a single displacement of the functional group. When it comes to the ping pong, there is a double displacement. In terms of a sequential reaction, again it is further divided into two types. One is the random order mechanism, which means there is an orderly, but that orderly happens randomly. On the other hand, in the same sequential manner, you have there is a compulsory order has to be there, which means what? Where both the substrates bind to the enzyme to form a ternary complex before the first product is formed. So that is called sequence. There is a sequence being observed in the bi-bi reaction. There is a sequence being observed in the two substrates involving reactions. So what is the sequence? The sequence is that both the substrate must bind to the enzyme, leading to the formation of the ternary complex before the first product is being formed. So this is called sequential reaction, which means there is a sequence being observed. The second type is the ping pong reaction, or the other word, the double displacement mechanism, or it is known as non-sequential mechanism. So which means there is no sequence being observed. So this is called ping pong. We shall see detail later. So when we talk about the sequential reaction, all the substrate must combine with the enzyme before the reaction can occur and products can be released. This is known as sequential reaction. So simple thing to remember is that for a sequential reaction, all the substrate, whether substrate 1, whether there are 2 substrates, or whether there are 3 substrates, or whether there are 4 substrates, all the substrate must bind to the enzyme before the product being released. So what is random order mechanism in this? So remember that sequential reaction can be of two types. One is the random order, second one is compulsory order reaction. So sometimes it will be referred as random mechanism. On the other hand, compulsory order mechanism will be referred as ordered mechanism. So don't get confused. So in such reaction, as I said, the group being transferred X is directly passed from A. A is what? A is one substrate, is passed to a second substrate called B, yielding to the product P plus Q. Right? So there is a single displacement of functional group X. So this is called single displacement reaction. So this is the reaction where representing a sequential reaction. So you have enzyme binding to the first substrate called AX. Now remember what is X? X is a functional group present in the substrate A. So don't misunderstand that AX is a double substrate. It is a single substrate. E combining to AX leading to a EX formation. On the other hand, there can be E binding to B, but B does not have a functional group. So this will form to EB. Now, to this EAX, second product or second substrate B can bind, leading to the ternary complex that I talked about. On the other hand, you have EB. To this, the first substrate AX can bind, leading to tertiary complex. So both these mechanisms are possible. Either it can go through this way or it can go through this way. But in both the instances, the final product of the first part is the ternary complex formation. So you have ternary complex being formed, AX plus T. Why ternary? Three components are involved. Enzyme, substrate 1 that is AX, and substrate T. Now, this ternary complex that we talked about, from here I said the functional group X will be transferred to that of B. So there is a transfer happening here, correct? So it still maintains a ternary form, and then from here there is a release of Bx. Now Bx is what? Bx is another substrate, the second substrate. To this substrate the X functional group is added. Now this will leave the Ea free, and from Ea there is E plus A. On the other hand, you have A being released from this ternary. As a result, you have E plus Bx. So these both the instances, that is this and this, can be also referred as P and Q. So here there is a product being formed and the enzyme free enzyme is retrieved back. So what is important is that there will be a two binding site on the enzyme. One for A and AX, another one is for B and BX. So this is what mentioned here. So you have a two binding site. So one binding site is here and the second binding site here. So this is number one and this is number two. Similarly, when it comes here, this is one binding site, another binding site is here. So depending upon where the functional group is attached, so corresponding to that binding site, the substrate will go and bind. So this we talked about so far is the random order mechanism. Now we move on to study about the compulsory order mechanism. What is compulsory? I told you compulsory means there is a sequence being formed, which means it is not just randomly where the substrate come and bind and go, but there is a sequence being formed and there is an order being formed. So what is important? In a sequential mechanism, the order of binding and leaving is compulsory. Okay, so this is important. The order of binding to and leaving the enzyme is compulsory. For two substrate reaction, a ternary complex will be involved, like we have seen in the random order mechanism, and the precise order must be specified. So example for here is E plus AX leading to EAX. Now addition of B will lead into the ternary complex. Now here the transfer of functional group into B, X is transferred to B, and the release of Bx, and eventually falling leading to the product formation, and thus results in the product formation, leaving the enzyme free. On the other hand, if it binds to the E plus B, and the sequential reaction will happen in this manner. So what is important is that there is an order of binding and there is a manner in which there is a release or leaving from the enzyme. What leaves from the enzyme is also important. So the enzyme will have a separate binding for A and AX and B and BX. So this is I already explained to you. In a sequential reaction, it can be classified into that of a compulsory order, which means order of substrate addition to the enzyme. There is a compulsory order which is said to be an ordered mechanism. So compulsory order can be said as the ordered mechanism. On the other hand, when there is no preference for the order of substrate addition, which means when substrate is being added to the enzyme, when there is no preference, a particular preference, like first substrate should come now and second substrate should come now, when there is no particular preference, and that mechanism or that reaction is known as random mechanism. So ordered mechanism versus the random mechanism, both fall under sequential mechanism. So in the compulsory order mechanism, the binding of the first substrate is apparently required for the enzyme to form the binding site for the second substrate. This is very important. So that is why it is called the compulsory order mechanism. Why compulsory order? When the first one binds to the enzyme, this creates a binding site for the second substrate. Whereas in the random mechanism, both the binding sites are present on the free enzyme. So this is the major difference between compulsory order and the random order mechanism. So Cleland shorthand notion. This is explained how the different substrates come and bind and different substrates leave. So you have the enzyme, free enzyme, with the kinetic K1, kinetic constant K1, the substrate A comes and binds, and you have the equal chance of the reversible reaction. Okay, so with the kinetic constant K minus 1, because it can be falling back after binding, EA formation may lead also to the reverse reaction of E plus A with the kinetic constant K minus 1. But it is leading to the, but now if we consider that E binding to A leading to EA, then it lead into the binary production, that is EA, and now you have K2 and K minus 2 as the kinetic constant with the B appearing inside, and result the ternary complex E and AB, leading to the formation of E plus Q, which is again a ternary complex with a kinetic constant of K3 and K minus 3, and leading to the release of the product number 1, which is P, with the kinetic constant K4 and K minus 4, and leaving the product EQ, then with the kinetic constant K5 and K minus 5, leading to the release of the second product E, resulting in the retrivation of the enzyme E, free enzyme. So the enzyme is represented in the horizontal line according to the Cleland shorthand notion, and successive additions of the substrates on the release of products are denoted by vertical arrows. So this is what we mentioned, what comes and what goes out. Enzyme forms are placed under the line. These are the enzyme form. So this is an ordered by reaction, right? Ordered by reaction. So where there is a regular order being observed. So many redox potentials, that is NAD plus and NADPH, requiring dehydrogenases, follow the ordered by mechanism in which coenzyme is leading to reactant. Now, what is the random by? This is the orderly, compulsory order. You can see the compulsory order here. So, where is A coming, B coming, then B leaving, and Q leaving. If you remember, initially I said before the first product being formed or first product being released, both the substrate must come and bind to the enzyme. That is the requirement for the ordered by reaction. Second one is the random by reaction. If you see by the diagram itself, there is no any particular order being formed here. So both AA being combining EA, for example, E, it can combine to A and forming EA, and to this EAB can form, and eventually another way of formation could be B and A forming and forming EB and leading to this ternary products converting into EP, and respectively the product being released PQ and leaving the free enzyme. So this you can go through by yourself because it's very easy. What I'm trying to say here is that unlike the ordered mechanism, here there is no any peculiar or particular ordered being formed in the binding of the substrate to that of the enzyme. That is most important. So some dehydrogenases and kinases operated through random by reaction. So this is example for this. In the previous cases, it is also dehydrogenases. In this case, there are dehydrogenases plus kinases. They also follow the random by mechanism. So if you remember initially that multi-substrate or the bi-substrate reactions can fall under two different categories. One is the sequential. So far we have seen what is sequential reaction. Now we move into ping pong reaction or the double displacement mechanism. Remember, difference between this and this is in sequential reaction only one displacement happens, one functional group displacement happens, whereas in terms of the ping pong there is a double displacement. So this is ping pong reaction. The mechanism in which one or more products are released before all the substrates have been added, this is known as ping pong reaction. So the major difference between the sequential reaction and the ping pong is in terms of sequential, both the substrates have to be bound to the enzyme before the first product being released. In terms of the ping pong, you have the first product being released before all the substrates could bind to the enzyme. So this is called ping pong reaction. So you have the free enzyme to which the first substrate A being formed, and there is a transfer of functional group from substrate A to that of enzyme. So the functional group is transferred to E, and here you can see already the first product being released before the second substrate comes to bind to it. So this is known as ping pong. Is the difference clear to all of you between the sequential and ping pong?
Yes, sir.
Father, can you repeat for the ping pong reaction?
Okay, I'll repeat ping pong. Now, what is sequential? We talked about sequential here, right? So in the sequential, both the substrate, that is A and B, have to be bound to the enzyme before the first product P being released. Is it clear?
Yes. Yes.
Yes. Now move to the ping pong. What is ping pong? In ping pong, you don't have to have the two substrate being bound to the enzyme. There is one substrate is enough binding to the enzyme. As a result, the first product A being released. Is the point clear now?
Yes, Father.
Okay. So there is a free enzyme to which the first substrate AX is combining, and after AX combining, the functional group X being transferred to E. As a result, it leads to the release of the first product A. And once the first product A is released, what is left out is the EX. And to this EX, the second substrate B comes to bind. The second substrate comes to bind. So as a result, B will combine with EX, forming to EXB. Once again, the transfer of the functional group, that is leading to ABX. If you remember, there is a double displacement. X is removed from A to E here, and again the X is removed from E to B. Okay, so there is a double displacement. As a result, you have the second product being released, that is BX, and leading the free enzyme as it is. So this is called ping pong. Now same way I have explained it another diagram, you can go through by yourself. This is as same as this one with the kinetic constant being present. So a functional group X of the first substrate A, right? So where is functional group? Here is the functional group with the first product AX. Sorry, first substrate. AX is the first substrate with the functional group X is displaced from the substrate to the enzyme. I explained to you to yield the first product P and the stable form F in which X is tightly bound to the enzyme, which is known as ping. This is known as ping. The first part of the displacement is known as the ping, and the second stage, the X is again displaced from the enzyme to the second substrate B to yield the second product Q, thereby generating the original form of the enzyme, which is known as pong. So that is why the reaction is known as ping pong. So you have the displacement first happening here and the second displacement happening here. As a result, you have the free enzyme being formed. So this part is known as ping, and this part, the second part, known as the pong. Am I clear to you?
Yes, Father.
All right. So this
Yes,
is known
sir.
as, yes, some doubt? Can I
No, sir.
proceed? Kiral, can you respond to me? Can I proceed? All the others?
Yes, sir.
Yes, sir. Yes, Father.
So
Yes, sir.
this is called double displacement. Now, note that ping pong bioreaction substrate A and B do not encounter one another on the surface of the enzyme. Many enzymes like amitripsin, transaminases, and flavoenzymes, they work under this particular mechanism called ping pong mechanism. Now, we are coming to another discussion in this multi-substrate. If you remember in the unisubstrate, we talked about the rate of reaction. We talked about kinetic constant. So the rate of reaction is that which will be enabling us to give us what is the velocity by which this particular reaction occurs. So in terms of the single substrate, we have derived the MM equation. If you remember, V is equal to, you know what is the MM equation? V max divided by Km plus S. Similarly, we also need to know about the rate equation for the multi-substrate or the bi-substrate mechanism. So this is same manner as that of the single substrate, but here because of the two substrate and the multi-substrate being involved, there is a complexity being involved in establishing the rate equation. So the rate equation for the above prescribed bi-substrate mechanism is this. Okay, so this is the rate mechanism. This is same as that of the single substrate rate equation. If you remember, single substrate is also same, now only given in the inverted form. So V0 is equal to Vmax multiplied by S0 divided by S0 plus Km. Km refers to kinetic constant and S0 refers to substrate concentration. Vmax refers to maximum velocity. Now this can be simplified and can be given in this form. So you separate this one and you will get this value. This can be represented as the Line-Weaver equation 1 by V0 Km 1 by S0 plus Es. This is for a single substrate we know. Now we have to convert this into a double substrate. Now for a double substrate, this is how the rate equation will look like. Now once again, if you remember, there is an ordered and there is a random. So for both we are talking here about the sequential reaction. In the sequential reaction, how do you find the rate reaction? When we talk about an order, so here is what given the rate reaction. What is the rate reaction? 1 by V0 is equal to 1 by Vmax plus with a kinetic constant Km for the substrate A divided by Vmax A plus Km for the substrate B divided by Vmax B plus KSA and KMB. We will study about Ks is what? Ks is the dissociation constant for A. Remember, A has to leave the enzyme in terms of order. If you remember, after both the substrate bound to the enzyme, substrate A must leave or the product A must leave. So this refers to the dissociation constant and divided by Vmax substrate concentration of A and substrate concentration of B. So this is the rate reaction for ordered by reaction. Now, what is for random? Random by reaction. This is the equation. So the 1 by V0 and you can go through this. This is for the random. Remember, this is for the ordered and this is for the random equation. Now, this ordered by reaction and this random by reaction both look like 1 by V0 is equal to Km by Vmax 1 by S0 plus 1 by Vmax. What this will remind you of? This will remind you of the straight line plot, correct? So y is equal to mx plus c. Mx plus c. You remember this, dear students? Y is equal to mx plus c?
Yes, Father.
Yes, so it is a straight line, right? So what m will refer to? M will refer to the slope and c will refer to the intercept on the y-axis, right? So this also will give you a straight line. So we shall see how from here we are able to find out what is the slope and what is the intercept. So this we have studied about for sequential reaction. Remember, we are interested in finding out what is the rate equation for both for sequential as well as for the ping pong. So in discussing about the sequential, this is for the ordered by, this is the rate equation, and for the random, this is the rate equation. Now moving to the ping pong, this is the rate equation 1 by V0 is equal to Km plus M plus Km plus M plus 1 by V0. This is the ping pong. Now what is the Physiological significance, Vmax, as I referred, it is a maximum velocity. Kma and Kmb both refers to the kinetic constant as I separate. So that is the, as we know, half of Vmax. You remember what is Km? Km is the half of Vmax. The same principle applies here. And if you remember, the third component here, which is an additional component, is the Ksa and Ksb, representing the dissociation constant for A and B from the enzyme, which I explained to you earlier. So, differentiating by substrate mechanism. So, one can discriminate between the ping pong as well as the sequential mechanism from their contrasting properties in the linear plots of such those of the Lineweaver-Burk plot. So, from the Lineweaver-Burk plot, depending on the plot, one can differentiate what is ping pong mechanism and one can differentiate what belongs to the sequential reaction. So what is a diagnostic plot for ping pong? How do I recognize whether this particular reaction belongs to ping pong or not? So if you draw a plot 1 by V0 versus 1 by A0. Now what is 1 by V0? It is the velocity for a reaction which utilizes substrate A as a substrate concentration, right? So 1 by A is refers to substrate concentration A at a constant B. Remember, there are two ping pong will involve two substrates. So you keep substrate B as a constant, and this will yield a straight line of slope with this and with the intercept of on the 1 by V0. So this is the ping pong reaction we have seen. This is the reaction for this. This will be the slope and this will be the intercept, right? So this will be y is equal to mx plus c. Now the slope is independent of b, and here you can see the slope km, which you have seen from here. This is a slope and this will be the intercept. We have the intercept here. The graph is drawn between 1 by v0 versus 1 by substrate concentration. So this is for ping pong mechanism. Now similarly, this is drawn for 1 by B, that is substrate concentration B, and here you have 1 by V0, similar kind of uh, slope and intercept can be formed with respect to substrate B. Remember, once again, we are still dwelling with ping pong mechanism. Now, how do we do a diagnostic for the sequential reaction? Remember, sequential will include two things. One is the ordered, another one is random, right? So this will be for ordered. This again will give you the y is equal to mx plus c, and this is for the rapid, that is the random. This also is equal to y is equal to mx plus c, and respectively, if you go through this, you have the slope here, and the graph between the ping pong will appear in this manner. If your graph comes to show in this manner of slope, then you know that this is a ping pong mechanism. And if you have the slope in this manner, okay, with an intercept moving across the minus axis, then you will know that this is the random mechanism. So this is what is for the mechanism or the plot for 1 by B0. So this is how we distinguish between the what is a ping pong mechanism and what is the known as the sequential mechanism. So I think this topic we shall take it tomorrow. Now, where do we heading to? Now we have to also prove by using two different mechanisms, that is the product inhibition. Another one is using the isotope exchange mechanism, by which we will be able to differentiate whether a particular reaction belongs to the random order or it belongs to the ordered sequential mechanism, which we'll see tomorrow. So for time being, what we have seen is that how do we differentiate? By looking at the slope, right? Whether a reaction belongs to the ping pong or whether reaction belongs to sequential. That we will be able to make out depending on the type of a slope that we get when we draw the plot. Okay, is the point clear so far? So tomorrow what we will do is that tomorrow session around 9 o'clock only we will have. So tomorrow session we will study about the inhibition and exchange studies to differentiate the random and the order sequential mechanism. Any clarification so far?
No, sir.
Okay, so what I would advise is that you read both Voigt and Palmer. So I've combined both of them to give you this lecture. If you read both of them, you will be able to understand. Okay, all right. So that's it from me, and we shall see tomorrow. Probably we can log on to the same link, and we can continue with the same link tomorrow. Or if there is a change, I will give you a different link. Okay. Stay safe,
Okay,
dear
sir.
students, and I hope that most of you would have been vaccinated by now. If any of you are not vaccinated, please don't take a fear and take courage and go and get vaccinated yourself and stay safe and all the best. Thank you.
Thank you, sir.
You may leave.
Father,
Yes.
uh, can you please tell us that like कौन सी date पर final exam हो सकती है, like at the end of the month?
I have no idea. You will have to ask the HOD for that. She will know about it. So ask Sudeshna madam and she'll be able to give you that perspective.
Okay, Father.
Okay.
Thank you.